Okay, good morning, ladies and gentlemen. Firstly, I'd like to thank Shao Zheng for inviting us to come to this beautiful country, Singapore, and to have this chance to present our research to all of our distinguished colleagues, friends, and students. My topic today will cover a series of EEG and ERP studies on emotion and executive function, which including attention, memory, conflict, and inhibition. I am going to divide my topic into 4 parts. The first, emotion and attention. Second, emotion and memory. The third, emotion and conflict. The last one, emotion inhibition. Okay, let us move on to the first part, emotion and attention. As for negative bias, as you know, we can say ENB. Emotional negative bias. Attention bias has it easier for emotional stimuli to catch one's attention than neutral ones that were mainly observed in high anxious normal person. Okay, during the trial of negative and neutral, as we can see from here, ENB could occur in several temporal stages, distinguished by early perceptual detection, late evaluation, and reaction readiness periods. Here is a typical target of Q target. It's a typical paradigm of Q target. We can see from here, we modify the paradigm by using threatening pictures like point at you with a gun instead of a flash. In high anxious group, reactions were speeded up and the pivot amplitude was enhanced when targets appeared of the same location as threatening pictures relative to non-threatening one, whereas in low anxious group, pivot amplitudes tend to enhance when the targets appeared the opposite location to threatening pictures. These results suggest that the attentional bias caused by threatening stimuli is able to modulate the visual inputs in early perceptual stages, and this mechanism is markedly influenced by an individual anxiety level. Here is a typical audible paradigm. From this slide, we can see that the P3 and the N1, I think which one is the slide one? Okay. Were larger in person with high anxiety than that in low anxieties group. These people tend to inhibit all types of disturbing emotional information. Okay, it is as emotional attention can occur in neonates. We tested 31 new babies aged less than 6 days. Fearful and angry spoken syllables dada and their corresponding non-novel sounds, and the non-novel sounds follow the envelope of each emotional syllabus. Okay, during baby sleep, we record ECG wave. Here we can see that the red color one is baby active sleep. The green color one shows the quiet sleep. We can see from this slide, they have difference between anger and fear during baby's active sleep. Okay, let us move on to the second part, emotion and working memory. As we know, emotion can moderate cognition. There is considerable evidence that the influence of anxiety on performance may be mediated by effects on working memory. From Bagley's working memory model, we can imagine that it has 2 systems. One is verbal processing, another is spatial processing. Here is a paradigm of 2-back task. In verbal task, participants instructed to remember the name of the letter, but ignore the location of them. Whereas in spatial task, the location of the letter was to be remembered by the participants. For ERP results, frontal LPC affected by negative emotion was different in the 2 types of tasks. Only the negative emotion selectively decreased prior to P300 of spatial tasks. For the location of brain activity, we use fMRI technique. The emotion activity is at the medial prefrontal cortex, whereas the cognitive activity is at the precutaneous cortex. We use the time-locked delayed matching to sample task to explore the possible brain mechanism of interaction between active emotion and working memory. These results indicate that the differential effects of negative emotion on spatial and verbal working memory mainly take place during information maintenance processing, which implies that there is a systematic association between negative emotion and a certain spatial retention. This is what we call cognitive process. We use ERP technique to investigate time course associated with suppression of negative neutral memories in a TNT paradigm in young normal. They have 3 steps of this experimental procedure. First, training. Participants were trained to associate with 48 Q target pairs. Second, varying EEG was recorded while participants only view the face on TNT periods. Pictures were framed in the green color queue for think trials and in the red color queue for no think trials. Last, at the testing phase, each individual was shown one of the 48 faces and asked to describe its associated pair. Okay, for ERP results, PNT conditions show significant differences in early P1 and N2. Contrastingly, negative and natural memories show significant differences in late LN and LPP, only in the NT conditions. Okay, the third part, emotion versus conflict. The first experiment, we often use selective attention of emotional conflict. It is an upgraded Stroop paradigm. They have 2 experiments. One is face determining, another is word determining. Okay, you can see from here that the main effect is at N170. Enhancement of task-relevant information and the suppression of task-irrelevant information begins at an early perceptual processing stage. There are different task design, like Stroop paradigm along with cognitive conflict, emotional conflict, and flanker tests. From this slide, we can see that the similar N2 and N450 effects are both at Stroop and flanker tests. Okay, I'm sorry. Okay. For the results by using Lorenta, we can see that N2 and N450 are at dorsolateral prefrontal cortex. Here, the red circle is dorsolateral prefrontal cortex. The blue one is anterior cingulate cortex. They have interactions between cognitive and affective control. Okay, above all the research we have done above, we can describe cognitive and affective control model. From here, we can say that the black line stands for cognitive control. On the basis of it, we added red line stands for affective control, and the blue one, cognitive and emotional control. The last part, emotion inhibition. We usually use emotion go-no-go task paradigm. In this testing, you can go with pressing button when you see glad face, and you can no-go without pressing anyone while you see a sad face. Okay, we often use emotional stop signal task. Here is the go trials. The other one is stop trials. The perceptual processing of the emotion cue could attract further attention, which in turn could be used for executive control, thereby enhancing behavioral performance, such as through short stop signal response times. Okay, dual competition model was proposed by Pozova in 2009. These mixed results can be interpreted through a dual competition framework, whereby emotional content influence the competition between perceptual and executive control. They have two purposes of our study. Number one, to examine the hypothesis that emotion could modulate response inhibition via perceptual processing of emotion content. Second, to expect the emotional phases would evoke increased amplitudes or shorter latencies of inhibition-related components related to neutral phases. There are two hypotheses and expectations. Suppose one, emotion was related to task. Explicit emotion would promote behavior inhibition. Suppose two, emotion was irrelevant with task. Implicit emotion would block behavior inhibition. For the time limiting, I just explained experiment 1, effects of explicit emotional on behavior inhibition. It is a 2 times 2 times 2 design. That is expression times response times valence. Ground average ERP waveform for no-go and go trials showed us that the main effect of response type is the N1 and P3. And also, emotion effects were for the VPP and the N170. The response type times expression interaction had a marginal effect. Emotional expressions elicited larger P3 amplitudes than neutral expression in no-go trials. And no-go trial P3 latencies were longer for emotional expressions than for neutral expression. Thus, the P3 effect may reflect the impairment of inhibition by emotion content. Okay, I'm going to summarize. Hypothesis: 1, emotion value index of response of the task. The explicit emotion enhances the sensory representation of the inhibition signal, which in turn decrease the need to inhibition and increase the outcome monitoring inhibition. The interaction between emotion and the response type were found in the sensory representation stage, N170, inhibition stage, N2, and outcome monitoring inhibition station, P3. Since we still have subsequent experiments, 2, 3, and 4, but I don't think I have enough time to explain to you at this time, so I'm sorry. On the basis of them, we can describe 4 processing stages for emotion modulating inhibition tasks. Focus on explicit emotion and implicit emotion. So we can modify dual frame competition framework. The top-down control, but not down-top control, moderates whether the perceptual competition involves or not. Okay, at the end of my speech, I'd like to release a very simple advertisement. Our group has developed Chinese systems of effective model with 7 sets. Until now, we have published 300 plus articles around them, and more than 400 facilities have used all sets of them or some of them. Okay, at last, I want to thank my group members and my collaborators. Thank you. Thanks for your attention.